Okay, so how enzymes work, pretty much you have what's called an active site. Okay, so the enzyme is the big globular protein, and there's an active site of some sort. It has a certain shape to it. Remember I told you the lock and key example? This is how it works. All right, so the enzyme is the lock. All right, and our substrates are the keys. These have to be a very specific shape for them to actually fit inside of the active site. All right, so you have this gigantic molecule in this very small little area, but it's shaped in a way that only that substrate is able to fit in. And it's based not only by its shape, but also if there's hydrogen bonding or non-polar interactions or polar interactions. So like for example, I can have a very, this part of it, it could be very non-polar. And then our substrate right here, this part of it is very non-polar. So it interacts, where on the green part, it could be very polar, have charges, and on the enzyme right over here, it could be polar. So that's why it fits perfectly inside of it. Again, we're talking about the tertiary structure. All right, we are looking more specifically at what this site actually has. All right, and then it fits inside of it, and then it does its chemistry. Okay, so here, so once a reaction has occurred, it catalyzes it, it breaks up our products, and I have a reaction. All right, this is very specific. All right, so the two models have been proposed to explain the specificity of the substrate for an enzyme active site. Okay, so again, it's a lock and key model. All right, so states that the active site is a rigid cavity to react. The substrate must exactly match the shape. And again, not only the shape, but also its chemistry. So if I have non-polar over here, this should be non-polar. If I have very polar over here, this end should be polar. So it fits very specifically inside of there. Okay. The induced fit model states that the active site has a flexible shape which can adjust to fit a substrate. So you can also have another model. Another model says that this part of it is more flexible. It can actually fit things a little bit larger. But either way, it has to match all the chemistry. So if I have non-polar here, this has to be non-polar. This is polar, that has to be polar. Okay. So let's see, shape the active site before bonding. Okay, so you have an active site, glucose might be able to fit perfectly in here, and then it closes up onto itself, and then it does its thing, and it releases it. Okay, factors that affect the enzyme activity. So the activity of an enzyme is affected both by temperature and pH. So I just told you that if you increase temperature or pH, you deactivate or you denature. Let's actually use that word because that's the word that's used. Denature the protein. If the protein's denatured, it doesn't work anymore. Right? Increase in temperature increases the rate of the chemical reaction, so that could increase the rate, but we're talking a very small increase. We're not talking a huge increase. All right, our bodies, can range from 98 to about, let's say, 104, maybe, in order to be safe. 104 is getting a little higher. Anything higher than that, so that's why having a temperature it can be very dangerous. If your temperature goes too high, you start denaturing proteins inside of your body, specifically inside your brain. And that is going to have a huge effect on you. So that's why our temperature can't really go over a little bit higher. Actually, they have it right here. They say over 37 degrees Celsius. All right, they say that enzyme catalyzed reactions also exhibit increase in rate with increase in temperature until a maximum activity occurs. So anything above that, you are pretty much going to be in a lot of trouble. All right. So each enzyme has an optimum pH, so our pH you can't change. So it's just basically saying that enzymes can work in a very specific pH range and in a temperature range. And having a temperature way too high can cause a lot of issues. All right, let me do a calculation here real quick. So 37 degrees Celsius, just to give you an idea of what that is in Fahrenheit. So 37 times 9 divided by 5 plus 32. So that was weird. Let's see. So, so that is 98.6. Sorry about that. This is your body temperature. That's the optimum temperature that proteins work at. Okay. As you go higher, obviously, I start to get a fever. Once you get about that 104, 105 range, you're at a very high fever. If you start getting above this, especially the 107, 108, you're in real trouble because again, you're going to start changing the structures of your proteins and you denature the proteins. The reason your body has temperature is because if you have a foreign anything protein inside your body, it's usually a protein, a virus or a bacteria or something. It's mostly a virus. Your body immune system purposely increases the temperature of your body to denature these foreign proteins. So a temperature is good. We want a temperature when we have a virus because the increased temperature, our bodies are able to actually, still work at that 104, 105 range, but a lot of viruses can't. So the temperature actually kills a lot of those proteins in that virus. However, once you start increasing more and the virus is actually adaptable in that range, then and your body starts increasing 6, 7, 106, 107, now you're at the point where your body can't handle it anymore. And sure, the virus is going to die also, but you will too. All right, so having a fever is not bad. Nowadays we have drugs in order to bring our fevers down, but there's nothing wrong to have a little bit of a fever because again, it's killing a virus or a foreign invader inside of the body. Okay. Allosteric control is the binding of a regulator to the site of an enzyme that affects the enzyme ability in order to bind to a substrate. Okay, so I'll show you a picture about this. There is two types, negative and then there's an allosteric. Okay, I think I talked about a while ago that our body is pretty good at regulating the amount of compound inside of it. So if you have way too much of something, what happens is your body has a way in order to switch off making that molecule. So like for example, serotonin. If we have way too much serotonin in our body has a system in order to shut off the production of serotonin. And this kind of is a way to do it. Again, the production and the breakdown of everything uses enzymes. Okay, and if your enzymes are active, it will work. If your enzymes are deactivated, then obviously you can't make more serotonin or break it down, or things won't happen anymore. Okay, so what a negative allosteric control is where a compound of some sort, it doesn't matter what it is, attaches onto the enzyme not at the active site, but at a different site, and it alters its shape of the protein, thereby closing off the active site. So my active site is no longer open. I can't have a substrate go in there. I don't have a reaction. So for example, in serotonin production, I don't know if it's a negative allosteric, but I just want to use that as an example. Maybe what happens is when you have a lot of serotonin, it actually finds an enzyme and it bonds into it, and it changes the enzyme that it takes another compound and in order to produce it into a serotonin. So sometimes the serotonin or the molecule is trying to make, if you have way too much of it, it bonds onto the enzyme and it shuts it down so it can't make any more. Once this then pops out and I don't have a lot of serotonin, then it goes back into this state, and then I can go ahead and I can take a substrate and go in there and make more serotonin. Okay? A positive allosteric control is the exact opposite. This still affects the enzyme, but what it does is it does the opposite. It turns in an inactive enzyme into now an active enzyme. Okay, so it does the opposite. Here it's active. When it bonds, it deactivates it. Here is when it bonds, it activates it. All right, I can't think of an example of where that would happen, but I'm sure there are examples of that. Okay. Okay, inhibitors is kind of what I was talking about. An inhibitor is where you have an enzyme of some sort, and I have an active site, okay, and a molecule it goes in there and it does a reaction and blah, blah, blah. All right. However, an inhibitor is where it itself, it goes into here and it blocks out the actual compound that has to go in there. Okay. So a perfect example is when we talked about the SSIRs, the selective serotonin reuptake inhibitor, SSRI, sorry. So this drug actually goes inside of the enzyme that breaks down serotonin. Okay, so if I have an enzyme of some sort and I have serotonin, all right, when I get way too much of serotonin, all right, this serotonin, it goes inside of the active site and it breaks it apart. So I no longer have any of the serotonin. Well, what an SSRI does is that it's a drug, I'll put it as a square or whatever, this goes inside of here and it blocks that active site. So I keep producing more and more of the serotonin. These enzymes have been shut off by the drug, so the serotonin can't get reuptaked into it. And that's basically how this works. Eventually, this will come out of the active site, so I have to keep adding more of the drug in there in order to keep that inhibited. Okay, so a reverse inhibitor binds to an enzyme, but then enzyme activity is restored when the inhibitor is released. So that's when it comes out. And irreversible is when it covalently binds to it, and basically it destroys the activity of the protein. Okay, here's an example of penicillin as an enzyme inhibitor. All right, so penicillin is an antibiotic that kills bacteria because it irreversibly binds to the enzyme required to synthesize a bacterial wall. So it inhibits the protein that makes a bacterial wall specifically for bacteria. Okay, so penicillin has no effect on human cells because they are surrounded by a cell membrane. So this is why penicillin works, because bacteria do not have a cell membrane. So these are able to actually get into the actual cell, or a bacterial cell. It binds onto the the protein that makes a cell membrane. All right. Well, once it binds onto there, it's hard to see, but it basically changes its structure. It blocks that site right there, and eventually the bacteria, it can't multiply anymore. It basically stops its multiplying, and at that point your body is able to actually all take control and kill all of the cells. So it can't keep replicating itself. It's basically, it has a certain amount of people in the army. All right, so initially the army is replicating and then eventually it stops. It won't produce any more soldiers. Our body, however, constantly is producing billions and billions of soldiers while the bacteria has stopped. So eventually our immune system is able to all take control. Viruses, however, do have a membrane. So that's why penicillin does not work on viruses. Viruses, you basically just have to wait for your body to overcome it. All right, and again, by increasing its temperature, its fever, and things like that. All right, enzyme inhibitors. Reversible can be competitive and non-competitive. A non-competitive inhibitor binds to the enzyme, but not to the active site. So again, it has an allosteric effect. It changes its structure to it. A competitive inhibitor actually goes inside of the active site. Okay, so here is a competitive. Here's showing an example of a non-competitive, where this thing bonds to it. It has an allosteric effect, meaning it changes its shape to this, and then the thing, it can't fit in anymore. All right, and then a competitive, it's not showing you, but basically a competitive is where this thing competes at the same site as our substrate. All right, a zymogen is basically an inactive form of the enzyme. So when our bodies produce proteins, okay, or an enzyme, they might produce a lot of them at one time. Our bodies are constantly making them. However, our bodies might not need that many enzymes at one time. So we have these stored up. It's in storage. When our bodies don't have enough of a certain enzyme, our bodies, and the whole point of this is that it is the enzyme, except it has a little piece of amino acids on the tip of it, which prevent it from actually being active. What it does is it it gives it a tertiary and a quaternary structure that is the inactive form. Then our bodies have enzymes that actually will cleave that off when we need it, and then it recoils up into the active form. Okay, so it's called a zymogen, which is a proenzyme, meaning it's right before it becomes an active enzyme. It's the inactive form that can be converted into the active form. Okay, it's kind of like the reserves, all right, in the Army, right? We've got reserves that might not be active, but then at one point we might need them, and they're called upon, and then it goes into active duty. Okay. Enzyme levels. Okay, so here are tests. So again, a lot of you are going to be nurses or in the healthcare, and knowing what these enzymes do tell you something about a condition inside the body. So certain enzymes are present in high amounts in particular cells, which can be detected if these cells rupture or die. So if you analyze the blood and they have a high level of creatine, all right, that might imply that they're having heart issues. All right, if they are alkalotic or they have a lot of phosphorus in their body, it can imply either a liver disease or prostate cancer. And then if there's a lot of amylase or lipase in the blood, that can imply that you have a disease of the pancreas. All right, penicillin inhibits the enzyme of the cell wall. I think that this just goes more into what penicillin does. All right, we've already talked about that. ACE causes blood vessels to actually narrow. It constricts. All right, is the enzyme that it constricts our blood vessels, increasing blood pressure. So if you have high blood pressure, a lot of drugs are called ACE inhibitors. They inhibit the ACE enzyme, so your blood vessels do not dilate. I'm sorry, do not all constrict. Because if they constrict, you have high blood pressure. If you inhibit that enzyme, then your blood vessels stay open. Okay, so ACE inhibitors are given to those with high blood pressure to prevent the ACE synthesis from its zymogen. So a lot of these ACE enzymes stay in the zymogen and don't get called upon or activated. Okay, in fact, a lot of these ACE inhibitors are actually proteins found in snake venom. Um, uh, so it's kind of interesting. The way that a lot of snake venom works is that it actually, when they inject blood or their venom into the animal, it actually inhibits all of the ACE enzymes. And what happens is the animal loses its blood pressure because all of its arteries all open up and they no longer have blood pressure. So blood, it can't pump all throughout their body, and that's basically how they die. So since they found that, they thought themselves, well, what if we use that as a high blood pressure medication? If we give like patients a very, very small amount of it, it will actually allow their blood vessels to actually open up. And a lot of these drugs out there are actually amino acids that have been isolated, or proteins that have been isolated from snake venom. And it's actually an ACE inhibitor. So if you look into that, it's kind of interesting. All right. Let's see what this is. ACE cleaves these two pro parts of the enzyme, and then you have a very activated amino acid. All right. Okay, actually, you know what? Let me put this on pause. I'll see if I can find those for you because I find it very interesting. Okay, so I just wanted to show you a real example. So lisinopril is a very common ACE inhibitor that's given to people with high blood pressure. Here's a structure. Notice it is a dipeptide. All right, if you look, it's actually backwards. Here is the C terminus side, all right, and then a nitrogen, and then you have a C terminus in the end. So notice it's a proline. If you see our proline group right here, we have, actually, I think it's a tripeptide. Let me see real quick. No, it's a dipeptide. It just has this proline-like group to it, and then it has these two R groups to it. Okay, and it's isolated. It does say here somewhere it's isolated from the pit viper. So it says here that it's high blood pressure. It's an ACE inhibitor. All right. Let's see where it says it somewhere. Let me see if I can find it real quick. Here's some side effects. Mode of action. I'll put this on pause. All right, you can see right here in the history. So this drug, you can see. Let's see if we can see this. It shows you the structure. All right, it's not quite as complicated as lisinopril, but it's a very similar structure. Was the first ACE inhibitor. Is a function and structural analog of the peptide derived from the venom of a jararaca, a Brazilian pit viper. So there you go. So it's this compound was actually found, and then they altered the structure of it in order to get the actual one. All right. So kind of a real life example of what ACE inhibitors are, what they do, how they've actually used an animal or our natural environment to actually find these in our environment and say, well, hey, if it works like that in animals, if it opens up all the arteries or dilates arteries, why don't we try to use that in high blood pressure? And sure enough, they got a drug from it. Okay. So ACE inhibitors. All right, HIV proteases. Oops, sorry. An HIV protease is an essential enzyme that allows the virus in order to make copies of itself. So HIV, the protease. Let me get a pen here. All right, is the protein that it makes copies. However, there are HIV protease inhibitors that interfere with the copying to decrease the virus. All right, you can't really kill a virus, especially HIV. HIV is one of those that once it gets inside of you, it's in you forever, but you can limit the amount of it that there is inside of your body by these proteases. So you have a very, very small amount of it. There have been people, I have heard recently that people with HIV have been cured. They actually are able to get rid of it after years and years of using these, eating healthy, exercise, that your body can eventually, it can take over and eventually it can kill every single one of the HIVs. Or it can have such a low concentration that it doesn't show up anymore in your blood work. But any slight alteration, it can bring it back. Nowadays HIV is all under control. All right, so that is it for chapter 21. If you have any questions, please feel free to ask me.